there are three main methods to determine the number of contributors. There's a decision tree, machine learning, and a posteriori probability. For the decision tree method, depending on the lab, this could be a simple process or more complex. Usually it's pretty complex and very time-consuming. Also, since it relies on human interpretation of data, the results can vary from person to person or lab to lab, which means it can be less accurate. There are some software available that use this decision tree method, like Faster. So this can help speed things up and remove some of the human bias, but it can't consider all of the possible combinations. The second method is machine learning, and this one is less labor intensive for the analyst, and it's increased the accuracy, but the big disadvantages are that it requires a very large calibration data set in order to be accurate. And even then, it can be less accurate for higher numbers of contributors and can result in a bimodal distribution. The third option is a posteriori probability. And once again, this one is less labor intensive for the analyst, but unlike in machine learning, there is unimodal distribution, and it's got high accuracy even when there are a high number of contributors. And this APP method does require a calibration data set, like the machine learning, but it's not nearly as large. So now we're going to spend some time looking at an example of APP software called NOCIt. We will go through and define the signal categories. We're going to talk about how the software determines expectations. And then we're going to very briefly go over an example of the calculations. So with NOCIt, the lab needs to supply a calibration data set composed of single source samples in replicates and dilutions that have been generated and analyzed following that particular lab's SOP. Along with this, the lab also supplies the expected genotypes for those samples. And this calibration data set is going to give the software context about what allele calls and stutter and noise look like in the data from this particular lab. And so the definitions here are pretty standard. In this example, the sample has peaks at allele 12 and 15. And the reverse stutter would be one repeat shorter than the alleles, so that's 11 and 14. And the forward stutter is one repeat longer than the alleles, so that's 13 and 16. And the noise would include the peaks in the other allele bins. So in addition to those positions, the software also looks at peak height and takes into account decayed amplitude. And so typical casework samples are often degraded due to things like UV radiation or acidic conditions. And frequently there are certain materials that can co-extract with the DNA and cause PCR inhibition. So in these cases, the lower molecular weight fragments amplify better than the higher molecular weight fragments. And so you can see that with a ski slope effect in the electropherograms. And it can possibly lead to dropout of your larger alleles. So NOCIt calculates the decayed amplitude for the peaks and plots that value against the peak's allele height, as you can see in the graph on the right. So the software does this for all the peaks in the calibration data set, which you can see here as the little blue dots. Then the software will generate a model based on that data, and it'll use that to analyze the unknown mixture samples. Another thing the software needs to account for is stutter. And since the lab provides the expected genotypes for the samples, NOCIt can track instances of stutter and develop a model for the stutter's expected peak heights based on the parent peak height. And yes, NOCIt does it for both forward and reverse stutter. So here's an example of the forward stutter. So NOCIt also looks at how the decayed amplitude affects the noise peaks. But as you can see here, the noise is just kind of more variable, so the model gets closer to a constant value than it did with the other variables. Dropout is also calculated. Here we can see the probability of dropout based on the decayed amplitude. So there's a higher probability that a peak will drop out if the decayed amplitude is small. You can also see the probability that a stutter peak will drop out based on the height of the parent peak. So the probability is higher when the parent peak height is low. And the noise detection rates against decayed amplitude are a constant value since the noise is more random. So now that NOCIt has models based on the lab's calibration data, we can take a look at the calculation. NOCIt randomly samples the genotypes for unknown persons, the level of contribution for each contributor, how degraded each contributor is, and the dropout rate. The software does random sampling since the number of possibilities is huge when you have a large number of contributors. So we are going to go through a quick example calculation. Here we have an electropherogram with peaks at alleles 8, 9, 10, and 11 with the peak heights labeled on the allele labels. And we're calculating the probability of this peak pattern given the number of contributors and the other information. So we start off with assuming there's one contributor. The contributor is randomly selected to have the genotype 1011 and no dropout and no degradation. So this means that the peak at 8 must be noise, 9 is reverse stutter from the 10 is an allele, plus some reverse stutter from the 11, and the 11 is the other allele. So when the software applies the model, you get a low probability that 8 is noise and 9 is reverse stutter, a moderate probability that 10 could be an allele and reverse stutter, and 11 is just an allele. But so overall, the probability ends up being very low. Here is another single contributor example. But this time the genotype is a 9 with a dropout of the second allele and no degradation. And so once again, you apply the model, calculate the probabilities for each and multiply. And once again, overall, this one is very unlikely as well. This last example is for 2 contributors. So the genotypes and contributor ratio is randomly selected. So contributor 1 has dropout, but there's no degradation. And once again, you apply the models, calculate the probabilities, and multiply. And yeah, with this option, it's also very unlikely. So this process is repeated over and over again and sums up, all of the n equals 1 possibilities, n equals 2, and so on for all of your n values. And NOCIt will generate a report giving the overall probabilities for each one of those possible number of contributors that were tested. So now that we've talked about how NOCIt works, we'll take just a little bit of time to discuss the validation and kind of the use of the software. So the first thing to do in a validation would be to generate the calibration data set that gives NOCIt the context to develop the model. The developers have recommended that you use as many single source samples as possible with as much variety as possible in regards to the alleles and the stutter. If you don't have a ton of samples, but each one is pretty unique, there isn't much allelic overlap, um, then you might be fine. It really just depends on your particular samples and their profiles. But as an example, during testing and development, 50 samples were used, and they were amplified at 8 dilutions. If your lab uses multiple extraction techniques, then you'll want to make sure to include samples that have been generated by each one of those techniques. You'll want to have at least 5 dilutions that kind of mimic the range that you see in your lab. And you'll want to generate those dilutions in different ways, like diluting whole body fluids pre-extraction, but also diluting the extracts post qPCR. But you'll want to make sure that you're using one amplification protocol and one CE injection time per calibration data set. So if you have different protocols in your lab, you'll want to generate and validate multiple calibration data sets. And another thing to think about, you might want to include multiple lot numbers for your reagents and consumables, and you wouldn't want to include any negative controls. So once you have your data set completed, you can begin validating the model. And so you want to make sure to use a different data set for your validation than you used for building your calibration files data set. Basically, you can follow the SWGDAM guidelines for an internal validation. This would mean including samples to test the sensitivity, specificity, precision, as well as case type samples. You'll look at known single source samples and known mixture samples. You'll want to make sure to have different numbers of contributors at various contributor ratios and different levels of allele sharing. And then you'll also want to include different amounts of DNA so that you get some partial profiles and some interlocus and intralocus peak height variation. Now we'll take a look at the workflow for using NOCIt. The first step would be to bring in the calibration files that you put together. So here you enter in a name for this calibration set. You'll import a kit file. It's basically the panel file. It lists all the loci and the alleles that are possible in your data set. And then you import your calibration files. So that will populate the table below with all the file names, sample names for your samples. You'll also need to import a file that gives the expected profiles for each one of those samples. And if you include the identifier for the profile in your sample name, NOCIt can automatically match them for you. Otherwise, you're going to need to go in and label the known genotype for each sample manually. And then when you're finished, you can click Next. Then you'll want to select which kinds of stutter you'd like the software to model. And this would just be based on what you typically take into consideration when doing casework. Then you click Calculate so that NOCIt can calculate the models for all the parameters we've discussed. This will take a few minutes. Generally, the time will depend on how many samples you've included and the specifications for the computer you're running the analysis on. But when it's done, you will click Save so that you can save this calibration file for your mixture analyses. You can click on any of the parameters to review the plots and see the models that have been developed for the data. If you notice any anomalies, you can check your input files, make any necessary edits, and then you can rerun the calculation. Now you're ready to run some mixture samples. So to start, you're going to click the plus button on the upper left, and you're going to add new sample files. You're going to add the calibration file that you just created. You're going to identify where you want to export all of your reports. You'll enter in the maximum number of contributors your lab will report out. And then you can select to filter, which will provide a CSV file with the baseline filtered in the results folder. You can select the appropriate population for the allele frequency information, and then click Add Batch. Then you can make any modifications you need to for each individual mixture sample, like if you want to include a known contributor profile. Then the analysis is going to run. There's a status bar in the center, and as the analysis for each sample is completed, you'll be able to see a plot of the possible number of contributors and their probabilities. Here are 3 example samples that we ran. The first is a degraded single contributor. So you can see that the highest probability is for a single contributor, but because of the degradation, there is also a low probability of 2 or 3 contributors as well. And so this data can easily be misinterpreted when someone is visually evaluating the electropherograms, because that degradation can mimic a low-level contributor. The next sample was actually a 3 contributor sample, and that was also the highest probability determined by the software. The third sample was a 2 contributor sample, and that was also the highest probability determined by the software as well. I also wanted to note here that it's important to test more than the expected number of contributors. If the highest probability calculated by the software is also the largest possible number of contributors, then that indicates that the true number of contributors could be higher than what you tested. So, for example, in the center case, if we told NOCIt to calculate up to 3 contributors, and we got the highest probability at 3 contributors, you would have to report out that the highest probability was for 3 or more contributors, not just 3. There are PDF reports and CSV files exported for each sample. They list all of the settings and thresholds, the likelihoods and probabilities for each of the possible number of contributors, as well as that plot. You can also see how long the analysis took. For this degraded single source sample, it took about 21 seconds. Here are the 2 reports for the other 2 samples that we analyzed. The 3 contributor mixture took about 36 seconds, and the 2 contributor mixture took about 40. These run times are a huge improvement over how long it would take an analyst to manually go through a decision tree. So just to summarize, there are multiple ways to come to a conclusion regarding the number of contributors to a mixture. We went into detail on one of the a posteriori probability methods called NOCIt. NOCIt has gone through developmental validation and is currently being used in forensic labs. It gives accurate results with a unimodal distribution. It's easy to use and runs quickly. It gets calibrated to a particular lab's SOP, taking into account things like peak heights and stutter and dropout. You can condition it to known contributors like victims, and you can use the results in further analysis, like probabilistic genotyping mixture analysis. If you would like to learn more, the developers, Doctors Catherine Gergachek and Desmond Lunn, have a detailed recording at the RTI Forensic Technology Center of Excellence website. So feel free to check that out.